Cultural narratives often suggest that motor learning is a function of time, specifically thousands of hours dedicated to the repetition of perfect form. Biological data contradicts this model. As this schematic illustrates, research suggests that skill is acquired through a formula involving error density and post-session consolidation. The term muscle memory ignores the underlying neurochemical sequence. Physical movements are managed by the central nervous system, and the process of improvement is driven by specific signals triggered by failure. Observation of high-repetition learning tasks indicates that the rate of mastery correlates with the frequency of errors generated per unit of time. Automatic and rhythmic actions, such as walking or cycling, are managed by central pattern generators, or CPGs, located within the spinal cord. When attempting a new or complex movement, the hardware shifts to the upper motor neurons in the neocortex. These neurons manage deliberate, conscious, and initially uncoordinated actions. The signals from both the neocortex and the spinal cord eventually reach the lower motor neurons, which actuate the physical muscle tissue. Training involves a transfer of the computational load from the upper motor neurons to the automatic CPGs. Mastery represents the anatomical relocation of a task, where the burden of execution moves down the spinal cord to operate independently of the conscious brain. For a novice, the primary objective of a training block is the generation of a high volume of errors. Norman and colleagues identified in 2021 that these errors trigger a top-down processing signal from the frontal cortex. This signal creates a framing effect, making the involved neural synapses more sensitive to subsequent neurochemical changes. Consolidating a skill requires a high dopamine signal-to-noise ratio. A low baseline is punctuated by a spike when a correct movement is performed. Elevating baseline dopamine before training masks the spike, hindering the nervous system's ability to identify a successful repetition. Frequent errors prime the synapse for change, allowing the high-contrast dopamine spike of a successful attempt to select and reinforce the correct neural sequence. Once a practitioner achieves a success rate of 25 to 30%, the focus of the protocol changes. This 2018 study by Lappe and colleagues used piano keys to isolate which forms of sensory feedback drive motor sequencing. The results showed that subjects receiving accurate pitch feedback and those receiving a single uniform tone learned at identical rates. Learning only failed when the feedback was randomized. This indicates that the consistency of an external cue is more significant for progress than the specific sensory detail of the feedback. Practitioners can use metronomic pacing to provide this consistent external constraint. Anchoring movement to an auditory pacer increases the number of repetitions per minute. These external cues reduce sensory distractions, encouraging the CPGs to operate at higher speeds and accelerating the transition away from neocortical control. The cerebellum integrates visual field data with the body's physical movements. Visual inputs, including pitch, yaw, and roll, map directly onto the cerebellar cortex, which cross-references this data with peripheral proprioception. Because the cerebellum uses visual boundaries to calibrate the neural innervation of muscles, expanding the peripheral visual field signals the brain to adjust its internal limits on muscle extension. This sensory input triggers the release of inhibitory neural pathways, resulting in an immediate increase in the physical range of motion. While the eyes can influence the limbs, the effectiveness of mental rehearsal is limited, as shown by Gannison and colleagues in 2004. While mental rehearsal produced strength and skill gains of up to 35%, actual physical training resulted in a 53% improvement. Visualization activates the upper motor neurons, but it cannot provide the cerebellum with the proprioceptive data generated by physical tissue tension. A neural circuit cannot fully optimize without the feedback provided by actual physical resistance. The hours following an active training block constitute a sensitive phase for learning. Diane and Cohen identified a specific process that occurs in the brain during quiet wakefulness immediately following a session. Within the hippocampus, the successful motor sequence is replayed in reverse order. This reverse replay is a necessary part of consolidating the neural circuitry. Engaging in new, complex tasks or consuming high volumes of sensory information immediately after practice can disrupt this playback. Providing the brain with 10 minutes of sensory silence after training is required to preserve the neuroplasticity generated during the session. The protocol for rapid acquisition requires 3 stages. First, high-density error generation to increase synaptic sensitivity. Second, a high-contrast dopamine spike to select the successful sequence. Third, sensory silence immediately post-training to consolidate the sequence via reverse replay. High-frequency failure followed by sensory silence establishes the biological architecture of skill.